switch this around. It's not switching.
I am Paul, if you weren't here last week. And if you weren't here last week, can you raise your hand? Because we need you to introduce yourself. I'm Paul Brandel. I'm a former science teacher, high school level. And I started this class 16 years ago. We've been running it every semester because the university is good enough to send us people like Josh. And every week we get to update ourselves on some science feature. So could you introduce yourself?
Well, I'm at the opposite end of this spectrum here. I do not know much about science, so I just thought I'd come and learn something.
Fantastic.
I'm Glenn Hanson. I'm retired. I've worked at previously at what was Covance in drug development, and so.
Okay, one more.
Susan Johnson, recently retired from the university. Most recently, I worked at the Center for Health and Science.
All right, well, welcome. And this is Tom Zinnen, who's
Hello.
my co-coordinator. And if you have any questions about Plato or the class, let me know. Other than that, we'll turn it over to Josh. And he will introduce himself, where he's from. Feel free to interrupt him at any time with your questions as we go through the lecture. So Josh, tell us who you are and what you got to say.
All right. Well, thank you for having me today. This is actually a great opportunity. I'm used to talking to a lot of scientists in a really narrow field, so it's fun to actually talk about my science in big picture. So it feels like a rare opportunity. So probably a little more excited than I should be. So my name's Josh Mitchell. I am a graduate student at UW-Madison. I'm in the IPID program, which is the Integrated Program in Biochemistry. So it's two different biochemistry programs, School of Medicine and Public Health, and the college, and then CALS. I always forget what the acronym is. So originally I'm from Michigan, actually from West Michigan, right along the lake, which was just fantastic. And so my parents were from that area. Went to school at Grand Valley, which is, I think at this point in time, the third largest undergraduate university in Michigan. There's about 25,000 undergrads. It's not really any graduate programs, but huge amount of undergrads. And so I got into science there. Originally thought I was going to go and be a doctor and go into medicine, but I love the research too much. And so I was a technician for a while, where I actually met my wife, which was pretty delightful. She was an undergrad. I was a technician. We spent a year together doing research, and next thing you know, we were dating, and then shortly after that, we were married. And so she was actually in med school for one semester, and then same thing. She really liked the research. She came on board, and we both came to grad school together here, and that was in 2017. So this is the start of my 5th year here in Madison. So a Midwesterner by trade, I suppose. So what I want to talk about today is, I like to think of it as taking another look when we're talking about eosinophils and using some of these new technologies that have been developed over the past couple of years and decades, and really see the new things we can see, which is actually pretty wonderful about this. But to get us starting off, right, I want to talk about the lab. So I work for Dean Mosher, who's here on the left. So Dean Mosher is a hematologist by trade. He's the primary investigator for the lab. He used to be a practicing hematologist, but now he focuses just on his research. I also have Mats Johansson, who's in the middle. He is the senior lab scientist. I turn to him for a lot of different things. And of course, that's me on the right. I'm Dean's 26th grad student, and as he keeps saying, his last. So hopefully that's better than it is worse. And so in the lab, we work on eosinophils, but it's more appropriate to back up, right? Because what we really are dealing with in many ways is blood. Blood is a pretty incredible thing when you think about it. It makes up 8% of the fluid in the body, right? If we're talking by weight. And what's incredible is it kind of functions as its own organ. It has a lot of different functions that go along with it, right? There's a lot of nutrients that move in the blood, so it's supplying all the nutrients. Things like gases, your oxygen, your carbon dioxide, so it's moving waste out. It's buffering your body, right? So it's keeping things normal, that normal pH that we want to keep. And it also acts as a huge mediator of initial defense against anything foreign in the body, right? So it's something we talk about like when you have sepsis, right? When we have infection, leukocytes, which are this bottom part, play a huge role in keeping us healthy, at this first bit, right? If we're bleeding, for attacking anything that comes in if we get a cut, but also making sure that you get clotting, right? So the skin the clotting allows for this kind of formation so that you don't have this opening where pathogens or anything can come in. And so, right, blood is kind of this big picture, but what we look at a little more specifically are white blood cells, right? So I'm sure most people have heard of white blood cells at some point in time, whether that's from being in the hospital talking about white blood cell counts or anything else, science or research. And so what we specifically deal with, right, are going to be these cells at the bottom, right in the middle. Eosinophils, basophils, and neutrophils, which we call the granulocytes. Granulocytes, as you can see with this picture, there's these granules, kind of these little balls, which is this noxious little payload. So we like to work with these cells because these cells often will mediate a lot of different effects in the body as we go. And so to that point, right, our eosinophil, which I'll often reference as an EOS, right, just the EOS, we'll often abbreviate it, right? It's a granulocyte. You can see it's got all these wonderful granules, which are kind of in the red here. It's got a lot of different proteins tucked away in these little granules, which you can think of. They're kind of little cellular bombs. It's really what they are. So there's a lot of noxious stuff in there. But there's also some signaling molecules, which is really nice because the blood can also act as a signaling highway in a lot of ways. There's a lot of paracrine effect, right? Like a hormone might be produced in one area and move to another. It's going to get there through the blood. So there's a lot of signaling that happens in your blood, sending messages back and forth. And so these granules can have really big immunomodulatory and inflammatory effect.
Question.
Yes.
Looks like the nucleus is two globs with a bridge between them.
That's how they are. Yes. So eosinophils, right? So when we look at normal cells, we don't have a demonstration of it here. Normal cells will have a nice kind of oval circular nucleus, usually when things condense. Eosinophils and some of the white blood cells are very weird because their nucleus is shaped weird, kind of this horseshoe, bilobed is what it's called. And some of them, like neutrophils have segmented nuclei. So it's the idea that there's a bunch of little parts that all join together. So this is actually one of the unique features of eosinophils is this bilobed nuclei, this nucleus that is kind of horseshoe. And actually we'll probably see some images that'll show that bilobe feature to it. It's very interesting. Any other questions while we just want to check? How's the topical level? Are we doing okay?
Can you remind us what eosin is? It's a stain or something?
Oh, eosin, yeah. So that's actually what they're also named for as well. When you had initial histology, right? When you had the initial staining of cells, especially coming from blood, eosin was a type of stain, hence the name, right? It's stained really bright pink under the stain. So it's kind of funny, right? You get the naming actually comes from how the staining seemed to be, which is kind of funny. So one of the big things with eosinophils, of course, is they have these broad effects, but we don't really understand how that payload, right? How these granules, we don't really understand how that delivery mechanism occurs. How does the release occur? So that's really kind of the space I work in, is figuring out how this delivery happens in a number of different ways. And so one thing, right? What we thought eosinophils did. Ooh, it's gonna load, this is great. Okay. So originally what was thought was that eosinophils attack these parasites, right? In this case, it's a helminth. And so this is a fun video. I'm hoping it's gonna play well. So far, so good. Okay, so you see it, right? You see the little worm, you see all the wiggling cells, and then they immediately go on the attack, right? And they surround it. And the idea is that they surround it and they have this noxious payload, kind of these little bombs, and they'll deliver it, and it does this awful stuff to it, right? So I enjoy watching this actually more than I should. So, all right, we got to see it again. So you see them, they're squirming around. It's not like they recognize something's there that we don't want there, right? So they immediately surround it, and then presumably it's going to die relatively shortly thereafter. Right, so that's what we always thought. We always thought that eosinophils were related to these parasitic infections, right? Which isn't that they're not, but what we found with time is that there's more going on. There we go. There's more going on. So you see eosinophilia, which is essentially a high amount of eosinophils in the body, especially in the blood, eosinophilia. And so eosinophilia happens with a number of different disorders, diseases, and there's such things as eosinophilic asthma, eosinophilic esophagitis, EOE, and then also hypereosinophilic syndrome. So this is all a large amount of eosinophils. For eosinophilic esophagitis, it's in the esophagus, causes a lot of inflammation, very difficult to eat certain foods or to eat a normal diet. Eosinophilic asthma actually affects a certain amount of younger children as well. So there's a broad interest. What I think is kind of telling, right, is we often think of diseases that are in some ways are affecting us, right? And we've talked about sometimes you get tropical diseases, right? And you think tropical diseases are miles and miles away. What's interesting is things like asthma really occur in a lot of developed nations as well, right? Where you see red being greater than 10% of the population having asthma. So it turns out that eosinophils really do affect the entire world in many ways. And actually, as you see development in nations, industrialization, asthma seems to get worse, right? So breathing issues. And I think we've all maybe seen the images or videos during COVID, right? When economies kind of shut down and a lot of factories shut down, that certain areas in India and China had huge reductions in the smog and clarity in the air and what you can see. This kind of ties into some of this asthma, right? These allergenic effects where these little bits of dust and smog can cause disorders in your immune system. So this is a primary focus right now in hospitals, is of course these eosinophilic diseases and disorders. But there's a lot of growing interest in eosinophils in new areas, right? And so we have gut microbiome and cancers, right? So the gut microbiome is, you have your gut, right? But your gut is full of bacteria because the bacteria, much like most animals, help ferment your foods, break down your foods. We often thought that the body breaks that down. Actually, all of these bacteria in your gut help you do that in a lot of ways. And so there's a lot of crosstalk between your body and this huge amount of bacteria in your gut. And so maintaining a healthy balance in your gut is incredibly important. And so it's thought that eosinophils play some sort of role in this, whether that's at making sure that there's a nice thick mucous membrane so any of your bacteria can't attack your cells, whether that's the maintenance of that barrier, right? Making sure these cells are happy along this border, or when things get out of hand, taking care of bacteria that suddenly start coming in and being pathogenic and being problematic. So there's consideration that EOS help modify and kind of help regulate what's going on in your gut a little bit. The other major interest, of course, is cancer. Because cancer, there's the idea that you have tumors, right? But there's a lot of work going on right now in the tumor microenvironment, which is that, right, tumors can be relatively big, and there's a very specific environment that surrounds these cells. And so there's a lot of interest in how immune cells, such as eosinophils, can either be antagonistic, right, can hurt these cancers, or how are these eosinophils contributing to these cancers, right? So in green, you have cancers where eosinophils are beneficial, right? Oral cancer, breast cancer, gastric cancer, right? But you also have some in the red which are problematic. And we're not entirely sure which way eosinophils are playing this role, right? In lung cancer, it's problematic. Hodgkin's lymphoma goes either way. So there's a lot of interest in how do eosinophils, right? This delivery of this payload, this noxious part, is really interesting to us because that can help us figure out how can we better affect certain diseases and disorders associated with some of these pathways, especially the cancers. So one of the important parts, though, is that eosinophils, they got to be activated, right? They're not, when they're in the blood, they're just kind of floating around really nice, which is great. But they respond to stimuli, right? They need to be activated. And so once an eosinophil, white blood cell, is activated, what it does is it's something called extravasation, which is essentially it leaves the circulation, goes in between the cells that make up your veins, your arteries and all that, slides out, and then goes into the tissue, right? And so it's really when it gets into the tissue that it becomes important. Because at that point in time, it's following a lot of these gradients, it's following different signaling molecules, telling it to go to a particular place, right? Kind of the Goldilocks thing, right? Leaving bits of bread so you can follow it afterwards. That's what a lot of cells do in your body. They'll leave little bits of these different compounds to try and draw these eosinophils out. And so there's a number of different ways that you can see eosinophils get activated. In the lab, I really look at two of these activators, and these activators are called cytokines. So unlike some of the other activators, cytokines result in very specific activation and some shape change of these cells when they go from spherical to a little bit of something else. And so I look at IL5 and IL33, what they're called, there's these interleukins. So IL-5 is the big one, right? IL-5 is released from Th2 cells. So that's an immune cell that helps regulate your immune response. And it releases this IL-5, which leads to differentiation and proliferation. So essentially it makes more eosinophils and directs things towards eosinophils, makes more of them, also leads to this activation. IL-33 is an interesting one because IL-33 gets released when epithelial cells, right? Cells that are lining a lot of our soft tissue, when they become hurt, whether that's become pathogens, whether that's because of things like cuts, mechanical stress, when they open up, they release IL-33, the signaling molecule, leads to the activation of these cells. So I look at these specific activators and they kind of have these different pathways. So what does this look like, right? So when we have a light microscope, if you look at them under a microscope, I don't know, maybe about 1000x, right? So nothing too intense. What you'll see is that your eosinophils are these nice spherical cells right here, right? They're spherical. This is the nuclei. So there's no coloring here, right? So this is the nuclei. And these are all the granules, right? Kind of this textured area. It's pretty small, right? So we work in micrometers, which we call microns. So very small. So thousandths of a meter. So very tiny, right? Not visible to the eye. And then when you add an activator, in our case, IL-5, right? So we'll talk about IL-5 first. You see a total shape change, right? Spherical, we call this the acorn. And so what's really unique about this as well is that it separates kind of into two sides. You can almost divide it right down the middle, right there. Right here, this is the nucleus, right? That bilobe nucleus we talked about earlier, jams in the back, right? So party in the back. Granules are all up front. So this is kind of the bomb delivery, right? All the noxious stuff is up front. And the cell moves towards the granules. So it'd be migrating in that direction. Now, if you do IL-33, we call this the pancake, right? It's very flat. It's much bigger. So here's the nucleus, right? Nucleus part 1, nucleus part 2, right? Still lobed, still there. But in comparison to these, where it's very textured and it's more condensed, these flatten out a lot. And they don't move that much either when it comes down to it. They maintain, they're pretty stationary when it comes down to it. So we're inherently kind of interested in how do you drive these cells one way or the other, right? What's going on with these cells?
It looks like those granules are of different material, varied material. Are they really, or that's just a picture?
Oh, for the picture? Yeah, so, well, I mean, the truth is, yeah, so they're all tiny little spheres almost tucked in there. And for the most part, they're all pretty even. Each granule is pretty close to the other in both shape and size. A lot of that, the kind of the texturing is the light itself. It's almost glare is really what it is. It's biological glare a little bit. And so if you look at the cell on the inside, right, this is so, feel free to ask questions too, because I realize this is maybe where it jumps a little deep, right? Polarization is very important for these cells. And so there's a lot of intracellular features that become very important for these cells. And so once you get activation, there's this nuclei, right? The big blue part. This is the nucleus, jams into the rear, right? You have these wonderful, this wonderful array, kind of signaling highway and support highway called microtubules. We'll talk about cytoskeleton a little bit later, but I want to show this preliminary image, right? You get this kind of supportive cage that goes around the nucleus with signaling. You have all these receptors at the rear, which is very interesting, right? So all the crosstalk, right? All the telephone gets played back here. You got the granules, which stick all the way up front. You have another cytoskeletal protein, F-actin, which helps with motility and moves it forward. So inside, right, it really is these distinct compartments. Nuclear stuff, payload, delivery mechanism. And so we're really interested in how this distinction gets made and what intracellular features play into this ultimately. Because when it comes down to it, degranulation becomes really important. And degranulation is the delivery of these little noxious bombs, these granules. And so currently there's 3 primary ways in which we view these things as being, delivered and dropped off. One of them is that the whole cell, the whole granule just gets ejected out of the cell, exocytosis. There's another way where little bits of the bomb break off bit by bit and slowly exit the cell. That's the piecemeal degranulation. And the other idea is that the whole cell breaks apart and then all the bombs just scatter around, right? So in 2 cases, the cell's alive, 1 case it's dead, right? It's kind of a suicide delivery by cytolysis, which is a very interesting thought of what that looks like and what's the importance of that. And so in our attempt to understand degranulation and these mechanisms, right, we're interested in how the activation of these signaling factors, these cytokines, affects both the polarity and the morphology of these cells, where they go from these nice spherical cells to these very distinct morphologies. One's a little more modal, this acorn shape, one that's a really flat pancake that doesn't move much. And ultimately, what's maybe the difference between these two? Do they both release things in the same way? Do they release things a little bit different? And how do intracellular features play into that? All right. So getting into the meat of it, right? The cytoskeleton. Cytoskeleton is very interesting. And it's mediated really by what we call kind of three main categories, right? There's microtubules, there's actin, and there's intermediate filaments, right? So you want to think of it as kind of like a skyscraper, right? Cytoskeleton, that's the walls, that's the beams, that's all the metal support that goes in. It's also the hallways, it's the doors, right? Cytoskeleton plays all features. It's roads, it's support, it's a wild thing. There's entire journals dedicated to just the cytoskeleton, right? So this is a very interesting part in science. And so what we have here is this is a picture of the cell specifically stained, right, for each specific cytoskeletal feature, where blue is going to be actin, green is going to be these highways, these microtubules, and the red is this intermediate filament. So let's get into some of these, right? So cytoskeleton, support, motility, and traffic. That's really what you want to think of it as. It's very unique because it's kind of tossing in three different concepts in one, right? It's your steel girders in a skyscraper, it's your highways, and it's also going to be your wheels in some ways. So it's very weird, right, that it acts like this. There's three classifications of cytoskeleton that we look at. There's actin monomers. So actin's real small. And actin plays a lot in motility when it comes down to it. They're called microfilaments. They make up actually a lot of our muscle too. So they'll often interact with each other, as you can see kind of in this red, but they're very small, 7 nanometers, right? So we're talking real small here. And so these have a lot of contractional force, right? So you see this a lot with motion, moving things forward. In the middle, we get intermediate filaments. There's a specific intermediate filament we work with in eosinophils. It's called vimentin. So intermediate filaments are very structural. They add to not only the rigidity, but they also have a certain elasticity. So they keep things intact, right? So this is a great example surrounding kind of a nuclei where you'll see a lot of intermediate filament, which gives a lot of support, right? So this in some ways, it's almost like the spider's web, right? Keeps things intact, keep things together. And then of course, we have our last ones are microtubules. Microtubules, in respect to the others, are pretty big. 25 nanometers. So these are like our superhighways. So not only are they supportive, but they also act as signaling highways where you have these little motors, little biological motors that'll scuttle along them, going back and forth in both directions, dropping off different signals or different little packages, which is pretty incredible. So I like to look at these three things and see how do they change from unactivated to activated, whether with one activator or the other. And so what we have to do, of course, right, is that it'd be really nice if we could just put them in a microscope and just look at them, but it's never that easy, is it? Never is. So what we do is we do something called immunofluorescence. And so the way immunofluorescence works is that you get cells, in our case, you know, it would be eosinophils, but it could be any cell, right? You grow the cells, and then you got to fix them, right? You got to keep everything frozen. Because if things aren't frozen, if you're trying to get things to move, it's very difficult to see what's going on, right? Sometimes it's really nice to have snapshots. And so you fix it, you make it stationary. Well, then you got to get these dyes inside the cell, right? Because everything we want to look at is inside. So what you have to do is you kind of have to drill into these cells. We call that drilling in permeabilization, right? Where you use different buffers and different components to puncture these little holes so we can get stuff into these cells that are fixed, right? They're not moving, they're frozen. So you permeabilize, lets us get things in, and then you add antibodies, right? So I'm sure we've all heard of antibodies before, of course, certainly not in this context. Antibodies are very neat because antibodies recognize very specific parts of foreign things, whether antigens or sometimes even our cells, right? That's one of the issues with things like arthritis, right? Is where you can get autoimmune disease, right? It's when your body recognizes things. A lot of this is by antibodies. So what's really nice is we can design these fancy antibodies to target very specific proteins or features inside of a cell and tell us where they are. And so what you do is you use antibodies, you add these nice fluorescent markers to them, so that way we can look at them in a microscope. And then what we can do is then look at them under a microscope. So this is a relatively fancy little technique that's very convenient for us looking at a vast array of things in cells, sometimes at once, sometimes one at a time. How are we doing with immunofluorescence? We doing okay? Okay, generally doing good, good. So this is what immunofluorescence on these cells looks like. So what we've got going on are some eosinophils. So I activated them, right? I gave them a specific little nudge, one direction or the other. In this case, it was with IL5. And what you'll notice is it's brightly colored, right? So finally some color in here. Dark blue, that's the nucleus. The red, those are those little microtubule tracks, right? That's a little highway tucked in right around the nucleus. And this light blue, the cyan, that's one of our other cytoskeletal proteins, actin. So actin usually contributes to movement, right? Motility. So we find that at the front because that's the direction this will be going. So this is really nice, you know? We see these beautiful little highways going across the nucleus. We see this nice modal front where we have this other protein that helps the movement. And then we presumably see all these granules that are going to be tucked in here too that may or may not be surrounded by this protein, which was kind of curious to us, right? Why is a supportive protein surrounding these bonds, right? We have no idea why. So that's actually one of the interesting questions we have is what's going on here. So this was nice. But one of the problems is that, you know, when you look at something that's frozen versus something that's active, right? Active is inherently better. We prefer active, right? It tells you a lot about what's going on. And so we were able to obtain some kind of rare dyes. And so it just so happens that a lot of times, right, the cell essentially has to be frozen, not moving, dead. So look at it. But it just so happens that occasionally you can get some dyes that work with living cells and living tissue. And this was the case, right? So here's this big fancy dye right here. It's called siractin because of this terrible abbreviation, just like inalide, which is kind of fun to say. And so what we did is we looked at these cells live just to see what would happen. And so what was really interesting to us is that we saw a lot of things that we hadn't seen before, which is that when we looked at this, right, so this is just actin, now it's in purple compared to the light blue. What we realized is that, yeah, there's some at the, you know, at the very front of these cells, tucked away over here, but there's this really bright patch tucked away by the nucleus, right, in the rear of the cell. And we had never seen that before. And to be fair, we still don't know what's going on, right? So that's part of the fun, right? We're getting partway there, but we're still trying to figure out what's going on. So that's kind of wild. But it was really fun to see these cells live, you know, moving and wiggling around, because normally we don't get this opportunity. Most of the time, they're just stuck there. So this was really exciting to see how do things change when they're alive versus dead, and what might we be missing. And so this was really nice, right? It gives us a little bit of an idea that actin's doing some weird stuff that we're not used to. So we decided to look at another cytoskeletal protein, right? Personally, my favorite when it comes down to it, which are microtubules, right? So going back to that earlier image, right? We see these microtubules, they move across the nucleus. We know that there's a lot of crosstalk that happens right here at the very edge of the nucleus. And so we thought, okay, this has got to be interesting. We're getting little highways that seem to cross over, but nowhere else. Well, we had just seen with one of these live dyes that doesn't look the same. Maybe it'll look different with the other live dye. And that was certainly true. And these are fun. Let's see if it plays. Yes. These are my favorite because I often think of it, it's like an octopus in a bag, which is kind of a weird thing to think about. But if you watch this cell in particular, this guy right here, right? So we see these little networks, right? So you can see this bright staining that goes to the back. Those are our microtubules, right? These nice little highways that cross over. But you'll also see that as it moves in this direction, right? You'll see it would almost look like these arms extend out, reach, and drag it forward. Which, the funny thing is I could watch this all day, which is a terrible thing. I won't make all of you watch it all day. But this was an incredible thing because we had never seen anything like this before, right? We had never seen these little arms reaching all the way to the front and dragging the rest of it with it. So this was entirely new because a lot of these highways are really dynamic. And when you have a cell that's dead and frozen, you don't see any of that dynamic movement. You don't see it getting put together you don't see it getting put together and then falling apart. And so in this case, we get to see that. We get to watch these highways form and then collapse and then reform and collapse and also drag things forward. Totally new to us, really exciting. So this live imaging has been a really fun component for us because it gives us a totally new view of these cells.
So how long have you seen that? 10 years, 5 years?
About a year. About a year now. So this was totally new. We'd never seen it before. Nobody had talked about it, which is part of the fun thing with these cells is that eosinophils, they're like the little brother of the cells, right? They don't get a lot of attention. They're like they're more of like the middle child out of 30, right? They're like child number 20, right? You give them a little bit of attention, but you got other fish to fry, right? So they don't get a lot of attention with research, which is really nice for us because we get to play around with these cells and look at all this fun stuff. And so that was the same thing, right? We looked at vimentin, this intermediate filament. This is the stuff that usually is supportive. And so what we found is before you activate any of these cells, right, they're just floating around in space and moving around in your blood. When they're spherical, this protein makes these beautiful little cages, right? And so the red is kind of an imaging artifact, but if you zoom through these up and down, it's like a spider web that crisscrosses all over these cells, right at the very edge. So this is the support that keeps these things spherical and nice and tight, right? Very hardy. They're very hardy cells, which is also nice. So we'd never really seen this before. We had to use a totally different fixation protocol. We had to do all sorts of crazy stuff. But that's why I'm grad student number 26, because I'm the crazy one. And so we tried all these different things. We got that. Well, then we thought, what would it look like if we activate these cells with IL5 or IL33? What happens when you get these shape changes? Does the vimentin move around? Does it stay the same? And what we found is when you look at the red, is that the cells totally change. The vimentin, which was this nice little cage, collapses down. And it moves around this blue nuclei. You see it goes in the middle and it goes around the edges. Now, it's not perfect in all of these cells. Part of that is the way I had to prep these cells. Wasn't particularly fun. But you see that in both cases, right? IL33 over here, when they make these nice flat pancakes, the vimentin really collapses down. IL5 over here, same thing. Really collapses down to the nucleus. And so what we're seeing is this new visualization for how these cells are working. Because before we had a really simplistic view. Highways cross over from the very tip to the middle. They move around. There's these smaller proteins that kind of move them along. Okay, that was great. But then with a little live imaging and looking at some other things, we find out they're a lot more dynamic than they thought. These big microtubule highways in the middle, they not only are they the highway that crosses over from the signaling area to the middle, but they're also the arms that help these cells reach forward and move. We find that the smaller modal protein, actin, right, these filaments, we find that they're tucked up here at the back, which we still don't entirely understand. And we also find that kind of this purple vimentin, right, which was red previously, right, we find that vimentin actually seems to form a cage also surrounding this nucleus, which seems to suggest that this nucleus is really important for us, right, to have that retention, that stability. So kind of the roll bars in a car. And this is also important to think of in the big context, right? Because if you have these cells and they're floating around in your blood and suddenly they get the signal to move into the tissue, right? You got to make sure every part of your cell is going to survive. And you don't want any part of your cell to burst, right? Especially something like a nucleus, because that's where all the information is stored, right? That's the epicenter. That's got all the DNA that's directing things. So a lot of what seems to be going on with these different proteins is that they're trying to support the cell moving out of your blood and into tissue, which is very interesting to us, just to see where that's going. So this was fun, right? This was a good view, but we wanted to get a more in-depth view because there's limitations to light microscopy, right? So there's a lot of crazy math that goes into it, right? But as we all know, you can only see so much with your eyes. And going from there, right, a lot of times we use light microscopes. Light microscopes are really nice. But we want to go more in depth, right? So we're talking very small numbers here, 100 nanometers, right? Really small. But it just so happens there's a lot of fun technology in microscopes that have an even lower or have even better resolution and can see even smaller, or see better resolution and can see smaller units of measure, right? So 1/10 of a nanometer. So you're talking about like picometers, right? Which is pretty crazy, right? That's incredibly small. You're starting to look at things at an atomic level, right? So you can almost pick out where individual atoms are, which is pretty crazy. And a lot of this is because the wavelength of the light that we use for these things is different, right? Light microscope, well, it uses the light for the most part that we can see, usually somewhere for 400 to 700 nanometers. That's what we see. Electron microscopes, right? The electron has a very different wavelength, right? And so depending on how fast it's moving, you can tune that a little bit and you can make it really small. So the smaller the wavelength, the more precision and the better resolution you can get. And so that's what we're trying to do. And it turns out that doing CryoEM, which is cryo-electron microscopy, is also on a very small scale too, right? So what we do is we put our samples on these little discs, and right, they look nice right here, right, where I have this nice demonstration of what it looks like. But here's a set of tweezers, right, with that tiny little disc on it, right? And that's a thumb, right? So that's a thumb, and that's the disc we're trying to work with. It's like working with tissue paper. It's terrible, and it's made of gold. So you feel even guiltier that you have gold tissue paper that you're trying to very delicately move around. And so what we can do is, before with our cells, our eosinophils, we'd put them on different surfaces and then we look at them. But we really have to do the same thing with this. So if we want to look at things at very small resolution, we've got to prepare them on a proper substrate, right? We got to put them on the right thing. And that happens to be these grids. So these grids, they've got this carbon surface, you know, it's like graphite, right? It's just this flat layer of carbon that they sit on. This flat layer of carbon sits on a gold grid. So if you were to look at it sideways, you've got this gold grid where it's got these bars with holes in the middle, carbon that the cells sit on, and then there's our cells right there, right? They just sit nice on top. And then what you do is you embed them in ice. So you dip them in a liquid and then you freeze them. So what's really nice about this is that when you freeze these cells, they're stationary, right? They stop moving. But without some of the issues we had run into before, because with fixation and permeabilization, you're screwing around with these cells around. Like if you threw a car in a frozen lake, right? Nothing's happening with that car. It's not moving, right? But if you have a car and you didn't make sure it's not going to move at all, you might have to take off an engine. You might need to take off a steering wheel, right? So in comparison, it's artifact versus no artifact, right? Because if somebody looked at the car later, they'd say, well, there's no engine in here, right? And there's no steering wheel. But if you have it frozen in ice, well, it's not moving and everything's still in place. So you can look at things a little bit differently. It gives you more detail without having issues with things being missing because of how you have to prep it. And so what we did, right, is we went back to the drawing board because you got to make sure everything's going to work. And it turns out that our cells love to just crawl along these grids. So this is a single little grid square right here. It's 90 microns by 90 microns. So it's very small, right? Just a small handful of cells are floating around out here. And once again, so these are the cells live, right? Once you activate them, so this is a cell, it's unactivated, it kind of squirms around a little bit, but the rest of these are activated. They're very flat. The granules kind of move around and shuffle around a little bit, and the cells will move around a little bit. This little edge kind of moves backwards and gets dragged along. And so this is really nice because, like some of the other images we'd seen before, it all looks the same, which is delightful. It makes us really happy to see that. And then on the right, if you put it in an electron beam, turns out it also looks okay too, right? So this is the very edge of the cell. So this was nice. And we get into higher magnification at these points in time. So this is where we go into this is where we dive into higher resolution, right? Because you start really zoomed out and now we're trying to move on it. We're trying to see what's going on at these little levels. So what we did is we actually went to the Pacific Northwest. We went to Oregon. In Oregon, they've got this beautiful CryoEM Center established by the NIH, the National Institute of Health. And what they have these really fancy microscopes, which is kind of hilarious because when you look at them, they don't really seem that fancy. So all this work, right, all this subatomic imaging goes on one of these things right here. That's it, right? For anybody that is familiar with the general surrounding Chicago area when it comes to science, right? There's argon, right, which is a synchrotron. So all it does is it makes X-rays, spins them around in a circle, and then you can do X-ray crystallography, right? A different type of way to look at structure. That's a huge footprint, right? It's a whole government center. It's covered with barbed wire. It's surrounded by fence. And this is just sitting around in a room. So it's amazing to see how technology has changed too with how we can visualize and look at different things in science, where it's getting smaller and smaller and smaller and a lot more easy to manage. And so what we did is we had prepped some grids before, like the ones I had shown you. We shipped them to Oregon and we were able to get some time to look at these cells and this was our SPOC, as she was called, our scientific point of contact, which I thought was hilarious. And she was absolutely delightful and really helped us move this project forward and get some of this preliminary imaging. And so this is what grids look like when they're not good. They look completely dark, and that's because electrons don't like to shoot through solid ice. So these are the little windows that got broken out, right? So you see some of that. So real icy, pretty icy, but hey, there's some stuff over there. That's why it's highlighted in red. And then this is what it looks like when the ice is a little thinner, right? Where you can kind of see this grayish color. That's the carbon film, right? So this is looking at a really high resolution. So what we were able to do is there were a couple of areas where we could look at these samples. And so we decided to collect some early imaging and see what was happening. So this is an obscenely detailed and up close image that honestly, even when I look at it, doesn't make a whole lot of sense because quite frankly, I'd never seen anything up this close up, right? Which was really cool. And so it's kind of figuring out what the heck is going on. But what we'll notice is that there's some kind of random shapes in here, right? So there's kind of this big circular vesicle, right? So the idea is that this is kind of this big biological orb floating right here, another one right here. And if you look carefully, there's a lot of little small ones tucked in here, right? So if you zoom in, that makes it look a little bit bigger, of course. Here's our big orb. Here's our smaller orbs in the middle. And here's another one. We've never seen this in any of our other imaging. This is just wild. So, like, and this is also right 200 nanometers. So in comparison to the other imaging, this is like 1,000-fold magnified in, which is crazy to think about, right? That's going from meter down to millimeter, and that dramatic of a change at this level. So this is really crazy. It's figuring out what's going on here. What we do see, of course, is if we look in a couple of different areas, right? We talked about granules before, those nice spheres that I had shown earlier. Well, it turns out when you look at them really close, they look like this, right? They look like these dark spots. And that's because they're chock full of goodies. They have a lot of these proteins in there. And these proteins, when electrons hit them, they stop. And so that's why it's really dark, because we're using electrons as our way to see through things. So the darker it is, usually the more stuff that's there, the more protein in this case. So it happens to be really rich, right? It's really dense, which is good. But what was really weird to us, because it was totally unexpected, was this little thing right here, right? You look at this one, it's a nice dark circular area. But that one almost looks like an oatmeal cookie or something going on with lots of raisins. So we were really curious about that. Now, this one zoomed in doesn't look quite as good. It almost looks better zoomed out. So if you stand at the back of the room, it's excellent. So what we found, right, is you have these dark little circles in here, right? So instead of it just being this uniform dark patch, there's these smaller areas, what we call punctae, right? Where it gets a little bit darker and the surrounding area is a little bit lighter. And you can see that there's this nice border that circles really the whole thing. What we can also see is it looks like there's some borders that cross in between, right? Almost like these little membranous rivers that kind of divide it up. We had never seen anything like this before. And there's a lab at Harvard that does a lot of work. The guy's name is Peter Weller, great guy. And he looks at a lot of this stuff, right? He looks at eosinophils and he likes to look at granules and he likes to figure out how do the granules exit the cell. That's really his big thing. Well, we've looked at a lot of his work over the years. We've never seen anything like this. The first time we've ever seen this big glob where it looks like it's breaking up into little parts. And so we're kind of wondering whether we're stumbling across something that is purely biological, right? Something that you wouldn't see in any other case because these cells, until they were frozen, were alive, right? So instead of all these other issues with fixing a cell and making it stationary, this is the real deal, right? An instantaneous snapshot. So what do we think? Well, we think that these big cells kind of get dense in little areas, crush down a little bit, and might kind of pop out. Kind of like if you had a giant corn cob and you popped it into a little popcorn and it budded off. So we're thinking that maybe there's this whole other way that granular content, right? These bombs can be released. We think they might condense down and then spray out in these smaller sections. But we don't know, right? Because this was the first image. So we're working on building up a lot of this data. So right, this has been a really fun imaging component because it's given us a lot of thoughts onto how cytoskeleton, right, changes the shape, makes these cells so half of it is nucleus, half of it is not, and also gives us an idea of these granules. So this was a really fun part of the project. The last part is a little bit different because what we move away from the idea of doing a lot of imaging and we moved into something called proteomics. So proteomics is pretty cool. It's really focusing on proteins, right? So proteins in our genome, we encode for 20 to 25,000 genes, right? And these genes ultimately turn into proteins, things like cytoskeletal proteins, all sorts of things. But the complexity really grows from there, right? So there's 20 to 25,000 genes, but there's different ways these genes can be handled, different ways they can be edited. So that leads us to the transcript, right? Kind of this middle ground. So if these are the instructions, these are the secondary instructions, right? And so you go from 20 to 25,000 to 100,000. Okay, that's something. Well, then there's one more step that you can do, right? Which is called post-translational modification. So once the blueprints actually get turned into the little unit, then things get crazy, right? Because maybe your unit's blue versus maybe your unit's red versus yellow, and maybe one unit's smaller than the other unit, right? So even though we don't necessarily feel, even though there's a lot of genes, right? But the complexity and how those things get put together, it's kind of like Legos. It gets out of hand really quickly with what you can build. And so we're interested in this part too, to see what's in these cells. So post-translational modifications, right, are something you're inherently interested in, because we're able to look at proteins and see how many proteins are in these cells. But what we're also interested in is what do those proteins look like? What are weird things that are happening to these proteins that may be causing serious changes? So there's a lot of what we call post-translational modifications, which are ways that proteins get altered, right? Almost like different paint jobs, or maybe you add a mirror here, you add something, or you add a tail there, right? And so you have all sorts of things, but we like to look at one in particular, right, out of all of these, which is called phosphorylation. Phosphorylation is really easy to measure. And phosphorylation does a lot of different things in the cells. When you phosphorylate a protein, you have this wonderful little molecule called ATP, adenosine triphosphate, right? It's got all these little groups on it. And what happens is that one protein will pop off one of these groups, one of these phosphate groups, and stick it on a protein, which doesn't seem like much, right? It doesn't seem like adding just this tiny little chemical compound to a protein can do a whole lot, but it does a lot of different things. So adding or taking away that phosphate group can change how cells coordinate their activity. It can change the localization where proteins will be, whether that's in the nucleus versus at the edge of a cell. It changes how things talk to each other, whether a protein will interact or not. It can change the conformation, where something could be nestled up next to each other, and then it forces some part of it to pop out, what things will bind to it, and also how long a protein may stick around. So a phosphorylated protein may be really stable, and it might stay around for, hours, days. It could be that if you phosphorylate something, it immediately targets it for degradation. It's like it puts a giant target on its back and says, get rid of me. So we were inherently interested in that, to see how does phosphorylation change when we look at these activators. And so there's this whole big workup. So basically what we do is we get a bunch of cells from donors, right? So these are all patient purified cells, which is really cool about this project. So we get blood from people kind of like us, although usually they have greater allergies, because that gives us more eosinophils when we purify them. We split them in half. We do this with 5 people, right? Split the cells in half. Half of them are activated, half of them aren't. And we do that with IL5, right? One activator, the other activator, or we do it with both activators. And so what we did, right, is then what you do, right? You have your collection purification, you activate them, you explode the cells, you break them apart, you break all the proteins down into these smaller pieces, and then you give them some fancy labels, right? You give them a barcode so you can figure out what's what. And then what you can do is then you can run these cells through this machine that'll tell you what proteins there are and what changes have occurred, which is pretty nifty when it comes down to it. It gives you an insane amount of information. And so Professor Josh Kuhn, he is at UW-Madison as well. This is what his lab does, is they are amazing experts in proteomics. And this is Laura Muehlbauer. She's a friend of mine. She's a grad student. She's the one that put together our samples. And so what we looked at were 3 data sets, right? We did a comparison. You activate it with one thing, and then you leave them unactivated. You activate it with the other, IL33 versus unactivated. Or what's the difference between the 2 activators themselves? So we wanted to try and do a comparison, right? Figure out what are the difference between these 3 different states, whether you look at them from an unactivated state, or if you try and compare them against each other. What you get is a lot of information. So I will also say we are still working on this part. So this is kind of the fun part. So when it comes down to it, right, let's talk some details. IL5 versus nothing, right? So the acorn versus the normal cell right out of the blood. Turns out you find 6,000 proteins in there. It's pretty incredible. It gets crazier though, because you also find 14,000 phosphosites, right? You find 14,000 different places in which the protein has a single little change, right? Just this phosphorylation. And then we talk a lot about significance, right? So significance is statistically doesn't change dramatically, right? So we find 4,000 of those events are important to us, right? The middle one's actually not as rich, which is saying something, because we still find 4,000 proteins. We still find about 5,000 sites. And of those 5,000 sites, about 1,800 of them are significant to us. Same thing, our other data set comparing the two activators against each other, 6,000 proteins, nearly 17,000 phosphosites, 3,500 of them are significant, right? So when it comes down to it, right, we have 36,000 sites that we're trying to look at, and we're trying to figure out what about 10,000 of them, what they're doing. That's a crazy amount of work. And so none of us in the lab know how to work with this data really well. So it's been a learning experience for the last year and a half. It's figuring out what the heck do we do with this, right? Just this incredible amount of data, which is fun. Now you can sort this data in all kinds of fun little plots, right? This one's called the volcano plot. I think it could be called an Old Faithful plot too, because it kind of looks like a geyser going off. But basically what this is telling us is that if there's a lot of changes, right? If you see this site really important, it's higher up, right? So things near the top, those we view as being more important. And if you see a dramatic change, right? If you see the presence of the modification being, you see it a lot more versus seeing it a lot less, it moves in one direction or the other, right? So if you see a lot of it, it goes to the right. If you see less of it, it goes to the left. And so basically beyond that, right, there's a lot of color that's on here, right? There's a lot of gray too, which doesn't seem to be very, very important. But there's a lot of these dots that seem to be really important to us. And the higher you move into these upper corners, whether it's upper right or upper left, those are the ones that are really interesting, because that means there's a really dramatic change going on, right? That means the moment these cells see this activator, bam, something really dramatic is happening to these proteins here or here, right? And so this is just with IL5, this is one activator, and this is another activator. So what we find is we have these nice little pockets of proteins that seem to be really changing, having a lot of dramatic changes. So we're still pursuing a lot of that. If you compare the activators against each other, right? If you compare IL5 versus IL33, the acorn versus the pancake cell, what's surprising is that when it comes down to it, these cells have a lot of proteins that are kind of similar. So in the red and the blue, those are specific proteins and specific sites that more or less, when you look at the two, kind of line up with each other. So what we did certainly learn, right? And so this is the idea that a lot of these phosphosites, there's a lot of them that occur in one protein. So some proteins will have 8 sites that change on. Some have only one, right? 413 have just one. What was really interesting to us is to see that there's a lot of commonality between these activators. So even though the shapes are completely different, and we can see that the interior cytoskeleton is very dramatic, there's a lot that happens when you activate them that's the same between these two. And so what we've been doing is mapping a lot of these different biological features, right? There's a lot of signaling that goes on in these cells. So we've been looking at a lot of that, right? These different signaling cascades and pathways, right? Different ways that things happen. And so what we've generally found is that whenever you activate a cell with either activator, a couple of things have to happen. Cytoskeleton's always got to change, which makes sense. You see dramatic shape changes. Trafficking seems to be affected in both cases too, right? Things moving across things moving up and down these highways, that's got to happen regardless. Ubiquitination is a way that proteins also get modified. And so we find that modification happens in both of these cases too, right? You have to have these modifications. And then there's splicing, right? Now splicing, unlike splicing cables, the idea of splicing in a biological sense is that when you get a bunch of DNA, you can change some of the order of it, right? So there's a bunch of these different kind of segments in DNA. Some of them you'll cut out, some of them you'll leave in. No matter what, it seems important. You got to screw around with the DNA when you activate them too. But we also found that there's a couple of things that are pretty specific towards just one cell type or the other, right? There's these big signaling pathways, which ultimately in hematology probably have more importance, right? Is on the left, if you activate with IL5, JAK-STAT signaling goes way up. And so JAK-STAT signaling, there's some issues in leukemias, right, where JAK-STAT signaling becomes very important. IL33 follows a very specific signaling cascade called the NF-kappa B cascade. So what we find is that these activators, for as similar as they can be at times, they also have very specific profiles that they must follow, and that there's very specific signals, kind of calls that come in that direct it one way or the other. So all in all, right, what we've talked about and what we found ourselves in is that these cells have very dramatic morphological changes, right? As soon as they see an activator, very dramatic morphological changes where they go from acorns and pancakes, and the cytoskeleton inside helps direct those changes. The granules, when we take a really in-depth look, really getting down to this almost atomic level, we find that there's a lot more going on than we expected, right? There's a lot more vesicles in there, but there's maybe new mechanisms for release that we've never seen before. So that's pretty exciting for us. And then, of course, looking at the proteomics, right? It turns out when you activate these cells, although there's a common core set of features, biological processes that have to happen, turns out that you still have very specific commitments that they have to make to go one way or the other towards acorn or pancake. And that sums it up, right? So here's me with what used to be my lab. A lot of these people have kind of either moved along or graduated. There's me a couple of years ago. This is Valerio, a good friend of mine. He just recently graduated. There's Mats and Dean, who are still in the lab. That's Dean's wife, Fran. Doug was our manager for a while, and then this was an undergraduate student that was helping us out for a while. But also, these are all the people that have helped me out, which has also been a blast. Certainly my own lab and the motion lab, which had this terrible temptation to always point at things, right? There's the motion lab, so this is the lab that I've been a part of, right? And then there's the people that have helped us with the CryoEM, right? That in-depth visualization. Igor, I didn't talk about his stuff, but this is a totally fun, different part. And then, of course, the people that move me along in my PhD career, right? The thesis committee. And then, of course, this is where I get all the money, which is always kind of nice for when we're doing experiments. So that being said, if there's any other questions, I'm happy to answer or clarify anything as needed. All right, in the back, what do we got?
Can you tell why you chose IL5 and IL33?
Oh, sure, yeah. So one of the things that happens with IL5 and IL33, this is more of the clinical side of things, which is kind of interesting. At a clinical level, right, if you have upregulation of eosinophils, right, eosinophilia, where you have more of them in the tissues, oftentimes eosinophils going into the tissue, you have to have more IL5 or more IL33, right? That's kind of a hallmark of some of these disorders and diseases. So they're a great model to look at biologically because we know they're significant. But the other nice thing too is that you see very dramatic changes. It goes from a spherical cell to these changes, it's crisp and clear. And that's another nice part too, is that instead of guessing if something's happening, you definitely know if something's happening. So it's kind of a twofold reason. Clinically, they're significant, and also for us to look at, it's kind of a no-brainer of like, oh yeah, something's happening. All right, next question.
When it's in the acorn stage, you got the granules on one end, the nuclei close to the other, and then you have those receptors down there. What kind of things bind to those receptors? And what does it do?
So on the very bottom, or kind of the little nose that sticks out.
Yeah. There's little receptors.
Yeah.
Past the nucleus.
Yeah. So let's see how quickly. No, I'm gonna have better luck by. No, that's gonna be terrible because then it's gonna screw everything up. I almost want to see how far back. Maybe it will. So when it comes down to the receptor in the back, what's thought about for that? Probably actually won't be too bad.
Yeah, way back.
Yeah, there's I think I had one right about in the middle. Right there. So we're talking about this part, right here. Right there. Okay, yeah. The signals, right? So the extracellular receptors, right? There's a lot of crosstalk that ultimately happens, and that's part of it, right? So when these cells are either in the tissue or when they leave, that's kind of the little antenna that helps judge where things are going. So what happens if anything binds to it, it'll tell it, okay, maybe we're going in the right direction. So if there's a gradient of this cytokine, right, these activators, right? So if it's more concentrated in one area versus less concentrated, the receptors in the back will help designate which way it's going. So it'll sense if I move this way, there's less stuff. If I move this way, there's more, right? So that actually helps the kind of the motility, right? Chemotaxis is what it's called, moving up the signaling gradient. So that's part of it. The other thing is that different cell types and different things can actually bind to that. One thing we found is that, right, platelets, which help with clotting, which are also, funny enough, nice spherical cells when they're just traveling around in the blood, they'll bind to the back of that as well. And so there's some people in the field that believe that platelets binding to these cells actually helps them escape the vasculature, right? Move out of the veins and the arteries and the capillaries. And they think that you actually have to have this smaller cell bind to it, which then allows it to escape. Now, a lot of it is also, it's just going to be a lot of crosstalk too, right? If IL5 is in the area, that'll help it become maybe a little bit more modal, or maybe it'll designate a little bit of a shape change too. So it serves, it really is kind of like the telephone in the living room, right? Everything's got to come in through there, and it'll tell you whether, to check the fridge, turn the TV on, right? That's really how that works. So it's really cool. It's kind of all purpose.
So why are there elevated levels of eosinophils, particularly in the United States, do you think?
Oh, that's a good question. This gets into a curious case, right? There was an article in the New England Journal of Medicine about a year ago that caught my attention, because it was actually comparing two different kind of farming populations. There was the Amish population in the United States, and there was what was apparently the kind of the Amish equivalent in Europe. What happens, how they interact with animals is dramatically different. In the US, the Amish population really interacts very intimately with animals, right? So there's a lot more contact. Versus in Europe, they found that the animal interaction in the population was more diminished. What they looked at then were allergies between the two groups. And what they found is that the more you interact essentially with these animals, the less allergies you have. It's some of the same things they consider now with peanuts, with early exposure with children, is that early exposure to peanuts, while sometimes can identify whether there is an allergy, if you continue this early exposure, it actually helps reduce your kind of allergic reaction to peanuts themselves. Now, it's not bomb-proof with everything, right? But it does help. So there is kind of a question in the US, is that because, especially in things like cities, because we may be interacting less with kind of our outside environment, are we seeing increases with allergies, right? Where you have this, kind of inflammatory response. The other part that goes with that, of course, is a lot air quality, of course, right? So if you have a lot of particulates in the air, those are immediately going to get recognized in the lung as being foreign. And so you get a quick immune response. So the more particulates you experience, right, the more your immune system is going to react. And so those concerns in major metropolitan cities that if the air quality is poor, right, if you have a lot of vehicles, interior, more smog, is that you're going to get higher rates of inflammation. So the idea is very interesting, is essentially that, kind of the ups and downs of highly developed nations too, is that because you have a lot of industry, yeah, sure, that's really good. It leads to different processes moving forward in society, but it can also contribute to a lot of pollution. So this is kind of that pollution edge of things, where pollution really has these dramatic effects. It's a lot more pronounced too in areas that either are enclosed by mountains, like Mexico City, or places where you have really bad pollution. A lot of certain areas in India and China, you also see terrible air quality, where not only are they masked all the time, but things like asthma are just off the charts. It's terrible. So you see a lot more of these kind of airway diseases in those cases. So that's actually a lot of it. There's a huge part that's environmental, which is very weird.
So if you are really trying, potentially, if you know more about eosinophils, then one of the things that asthma, particularly like now in Beijing and other places, including Madison, where people are getting more asthma. Do you have a simple just research, or do you have the lab have a target, or how does that work?
Right now, a lot of it is basic research, right? Really going at a fundamental level of figuring out what's happening, because these cells aren't well understood. And so kind of the idea is that we're kind of doing the legwork so that clinicians can come in and say, oh, that's important. So there's a little bit of a tie, kind of the ending, right? Where I talk about proteomics. Proteomics is oftentimes where you see more of that targeted clinical response, where we can say, hey, when we have an activator, right? IL5 is a great example, because IL5 is very important usually for asthma and eosinophils moving into tissues, especially the lung. We could say, hey, when you activate these cells, IL5, we notice that these 10 proteins seem to be really important. If we're able to prevent these proteins from being phosphorylated in this case, right? One of the things we looked at, does that make the tissue inflammation not nearly as bad, right? So, but a lot of that is also the basic biological work, right? It's by us doing a lot of that basic biology, we can move it up the ladder. So currently, we're not doing the clinical work, but we're helping move that forward. Because there is actually, it's interesting to say that there's a COVID project in the lab right now. So it's kind of weird. We got COVID samples in the lab. So you always feel a little weird when you see the door, the little sign on the door that says COVID samples are in right now. And we've been looking at a couple of proteins, same thing, that seem to be either upregulated or downregulated, right? Either you see more of it made or it disappears when you get infections with COVID. So we're also working at that on a more clinical level of saying, if you replace some of these proteins, does it actually help people that have COVID? Or, is it easier for them to breathe? Or does it reduce how bad they feel? Things like that. So we're working a little bit of each space, but for EOS right now, more of it's basic. Yeah.
Anyone else? And next week we have Parkinson's disease, modeling and visualizing Parkinson's disease. So, see you next week.